We studied how diacylglycerol lipase disinhibits ventral tegmental area dopamine neurons during chronic nicotine exposure. When nicotine use increases, it enhances dopaminergic signaling, which promotes more nicotine use. That is where the factor of addiction comes into place. This happens via alteration of synaptic transmission in the ventral tegmental area. Now, how this happens? So, when nicotine is injected, 2-AG production increases and the release of GABA neurotransmitter decreases. We experimentally determined and inhibited 2-AG by DAGL, and this rescued nicotine induced area GABA signaling after nicotine. Enhancement of 2-AG, inhibiting 2-AG by degradation, recapitulates loss of GABA signaling. The results were that the excessive 2-AG signaling resulted in the loss of inhibitory GABAergic constraint of ventral tegmental area following chronic nicotine exposure. Let us start by discussing what we already knew about dopaminergic area activity and ventral tegmental projections before conducting the study. So, the mesocortical limbic dopaminergic area provides link between brain regions processing cognition and those controlling motor behavior. Control of these ventral tegmental area projections helps in seeking rewarding stimuli and retreating aversive stimuli. GABAergic signaling provides inhibition that gates ventral tegmental area and dopaminergic cell excitability. Loss of this inhibition, however, leads to pathological dysregulation of mesocortical limbic circuitry. ECBs or endocannabinoids regulate dopaminergic activity through retrograde signaling from DA cell bodies onto CB1 receptors. These CB1 receptors can be of two types, 2AG and AEA. They both function as endogenous CB1 agonists in the brain, however, have distinct pharmacological profiles in vivo. The enzymes responsible for endocannabinoid degradation are characterized using pharmacological tools, and evidence implicates endocannabinoid signaling in the etiology of nicotine addiction. Chronic nicotine exposure selectively enhances nicotine induced increases in ventral tegmental area 2-AG formation. We hence study the contribution of this effect to aberrant ventral tegmental area dopaminergic cell excitation after chronic nicotine exposure. And we found that sensitized nicotine induced 2-AG release strongly correlates with the loss of nicotine induced GABA release. Let us now discuss the procedures and setup of the study briefly. Studies involving male Vista rats were conducted in accordance with NIH guidelines and as approved by the Scripps Research Institute Institutional Animal Care and Use Committee. Nicotine administration procedures. For violational nicotine exposure, SA rats were trained to operantly respond for IV nicotine under a continuous schedule of reinforcement in daily 2-hour sessions using previously published procedures. Neurotransmitter measurements. Quantification of neurotransmitters in dialysate samples were performed by LCMS-MS using established methodology. Gel-based competitive ABP experiments were performed as described as well. Electrophysiological recordings were taken, and for statistical analysis, microdialysis analysis significance was determined by repeated measures ANOVA for effects of group nicotine and group X nicotine interactions. Let us now discuss. Our first result indicated that chronic nicotine exposure impairs nicotine induced GABA release in the rat ventral tegmental area. Second result assessed how CB1 influence on ventral tegmental area GABA signaling is not altered following chronic nicotine exposure. The effects of the CB1 receptors ligands on SIPSCs did not differ between nicotine naive and chronic nicotine exposure ventral tegmental neurons. Our third result was about determination of the 2-AG influence on nicotinic induced alterations in ventral tegmental area GABA signaling, which required manipulation of 2-AG biosynthesis. We used ABPP assay and DAGL directed probes to evaluate the potency and selectivity of a series of 1,2,3 triazole ureas for inhibiting the 2-AG biosynthetic enzymes DAGL alpha and DAGL beta. Our results demonstrated that KT172 and KT185, they both serve as paired chemical probes for selectively modulating 2-AG metabolism in the rat brain. Our results also demonstrated that DAGL inactivation restores nicotine induced GABA release in rats who have a history of nicotine exposure. We tested if inactivation of DAGLs could restore nicotine elicited GABA signaling at the ventral tegmental area dopaminergic synapses in slices from chronic nicotine exposure rats. Slices were superfused with KT172 or KT128 but had no effect on baseline ISPSC's characteristic. Furthermore, our results also indicated that attenuation of 2-AG clearance blocks nicotine induced GABA release in drug naive rats. Loss of nicotine induced GABA signaling observed in the ventral tegmental area of chronic nicotine exposure rats can be reproduced in naive rats by inhibiting 2-AG clearance with inhibitors of ABHD6 or MAGL. Lastly, our results also indicated that nicotine self administration is reduced by DAGL inhibition. Discussion. So, we see how through the use of selective inhibitors of DAGL, ABHD6, and MAGL, the present findings reveal that enhanced on demand 2-AG formation curbs the inhibitory control of nicotine induced ventral tegmental area cell excitation following CNE. Loss of this inhibitory feedback mechanism contributes to increased ventral tegmental area sensitivity to nicotine and other stimuli, which results in sensitized dopaminergic release in the nucleus accumbens and increased motivation for nicotine intake. Nicotine induced increases in VTA 2-AG formation likely contributes to the well documented CB1 receptor influence on behavioral and physiological response to nicotine. In VTA, CB1 receptors are thought to dynamically regulate GABA release at dopaminergic synapses, allowing the possibility that 2-AG formation dampens GABAergic constraint of DA-cell excitation. A direct test of our hypothesis requires the suppression of 2-AG formation, although studies of this type have been limited by a lack of selective inhibitors of 2-AG biosynthesis and suitable biochemical assays to validate inhibition of endogenous DAGL activity. To address this, we evaluated the efficacy of DAGL selective 1,2,3-TU inhibitors for use in the rat brain using a tailored ABPP assay. We found that KT172 selectively inhibits DAGL enzymes in the exogenous brain proteome as well as in awake animals, and that primary off target of this inhibitor could be accounted for using the negative control probe KT185. Conversely, attenuation of 2-AG hydrolysis by either ABHD6 or MAGL in nicotine naive rats recapitulates the loss of nicotine induced GABA signaling after CNE. These findings support the hypothesis that nicotine induced increases in 2-AG dampen the GABAergic constraint of DA cell excitation. We find that functional influence of CB1 receptors on GABA release is unaltered by CNE and demonstrate that CB1 receptors antagonism robustly increases GABA levels and GABA receptor signaling. Nicotine and other drugs of abuse alter synaptic transmission in the ventral tegmental area through additional mechanisms that may be facilitated by the loss of local inhibitory signaling. Our results provide novel direct evidence that pharmacological modulation of 2-AG biosynthesis is a viable approach for evaluating the influence of ECB system on substance abuse disorder. Critique. In this study, we study the influence of ECB signaling in the brain. However, this influence of ECB is hampered by the lack of appropriate corresponding tools for selectively inactivating on demand ECB biosynthesis. Maybe these tools can be enhanced and the study could be further improvised by adding additional data. The study was done on lab rats, but we could also extend it to other animals and study them experimentally and extend the results to them as well. Instead of nicotine use, other drugs of abuse could also be used to test their influence on the ventral tegmental area.